Today we will be studying the structure of bacterial flagella. So now what is this flagella? When we hear about this, we tell that it is a locomotory organ. Apart from being a locomotory organ, it also acts as an antigenic structure for some of the bacteria. For example, Salmonella typhi. Salmonella typhi has a flagella which is antigenic in nature. Now let us see the structure, the basic structure of a flagella. It has three parts: the basal body, the hook and the filament. Now what is the basal body? Basal body is the component which is present, which is embedded inside the cell. So this consists of the basal body. The filament is the protein which is seen outside of the cell. It is made up of a protein called as fibrin. Now the filament and the basal body is connected to each other through a hook. Okay, now let us study what are the different components of the basal body. The basal body consists of rings. Now what are the different rings present on the basal body? It is divided into two rings. Outer ring, so these are called as the outer rings, and these are called as the inner rings. Now what does the outer ring consist of? Outer rings are of two types. One is the L ring and the other one is the P ring. L ring is the one which is present on the lipopolysaccharide layer, the outer membrane of the lipopolysaccharide layer. The P ring is the one which is present on the peptidoglycan layer. Now we have seen that the cell membrane and the cell wall is separated by a periplasmic space. So that periplasmic space, in the periplasmic space there is a rod. Okay, now coming to the inner rings, there are two rings called as the S and the M rings. Both these rings are embedded inside the cell membrane. The cell membrane, it is present inside the cell membrane of all the bacteria, all the flagellated bacteria. Now coming to, I can show you the different types of flagella here. If you see here this picture, you can see the filament. I told you the filament is made up of a protein. So this is the protein which is made up of flagellin protein, a globular protein. This is the hook and this is the part which can be seen outside. Now when we see the embedded, the basal body, you can see the outermost ring here, the outermost ring, and here you can see the outer membrane. Here you can see the outer ring, the second outer ring, that is a P ring, which is inside the peptidoglycan. You can see the peptidoglycan layer here. This becomes the periplasmic space, and these are the S and the M rings. Now these S and M rings are inside or clamped inside proteins called as the MOT proteins. So these proteins hold the S and the M rings. They are called as the MOT proteins. Now here in this picture you can make out the different types of rings. If you see here, this is the outer membrane, this is the outer membrane, and the L ring, the peptidoglycan, the P ring, the MOT proteins, the brown ones are the MOT proteins, and the S and the M rings. This is the cell membrane. The same picture is shown here as well. Now, what is the difference between the flagella of a gram-positive bacteria and a gram-negative bacteria? If you see, in gram-positive bacteria, there is absence of outer membrane, that is there is no LPS, there is no lipopolysaccharide layer, and hence even in the bacterial flagella, if you see, this is the bacterial flagella of gram negative. This is the bacterial flagella of gram negative, that is, you can see all the four rings, the L, the P and the SN and the M rings. But if you see in gram positive, you can see there is only peptidoglycan layer, absence of LPS, and hence you can see only the P ring, there is no L ring, and you can see the SN and the M rings. So there is no L ring in gram positive bacteria. The same thing is represented in this figure as well. You can see one ring here present in the cell membrane and the other ring here present on the peptidoglycan layer, the P ring. The same thing, this is in gram negative bacteria. You can see the presence of all the four rings here, and of course the hook and the filament is clearly seen. Now there are different bacteria with different arrangements of flagella. So they can be named based on the type of bacteria and type of flagella, number of flagella and the arrangement of flagella. Let us see each one in. If the bacteria do not have a flagella, or they are non-motile, and those kind of bacteria are called as atrichous. Example, Klebsiella pneumoniae. Coming to, if there is a bacteria with only one flagella at one end, then it is called as monotrichous, because there is only one. Mono means one, so it is monotrichous flagella. If there is a bacteria with tufts of flagella in one end of the bacterial cell, then it is called as lophotrichous. Example for lophotrichous is Bartonella bacilliformis. Bartonella bacilliformis. If the bacteria has one flagella each on the either sides, either polar ends, then it is called as amphitrichous. Okay, or tufts of flagella in either sides of the polar ends, then it is called as amphitrichous. Example for that is Spirillum serpenum. Spirillum serpens. Spirillum serpens. Now there would be some bacteria who possess flagella throughout its body. Okay, the flagella is distributed throughout its body, then such kind of bacteria we can call that as peritrichous. Example for that is E. coli. I'll just revise once again. If the bacteria do not have any flagella, they are called as atrichous. Example, Klebsiella pneumoniae. If the bacteria has only one flagella at one polar end, it is called as monotrichous. Example, Vibrio. If the bacteria has tufts of flagella in one end, many flagella at one end, it is called as lophotrichous. Example, Bartonella bacilliformis. If there are flagella on either sides of the bacteria, polar ends, it could be single or tufts of flagella, then it is called as amphitrichous. Example, Spirillum serpens. If there are flagella which is distributed throughout the body of the bacteria, then they are called as peritrichous. Example, E. coli. Thank you.